Today we'll discuss the new drug development, NDD, that is a new drug development. In that we'll discuss the drug discovery. Most of the new drugs are discovered and identified through one of the following approaches. In the drug discovery, random screening is one of the approach. Now what is meant by random screening? Random screening is a sort of blind hitting procedure where we don't know anything. It's a blind hitting procedure where new chemical entities like natural or synthetic new chemical entities, they are subjected to the battery of pharmacological screening procedures. New chemical entities, they are under pharmacological screening procedures. They are designed to explore different types of biological activity. They are designed to explore different types of biological activity, and that test includes pharmacological screening procedure include what it includes. It includes the animal behavior, studies on the animal behavior, animal model of the disease, isolated tissues. These are the test includes. And the important thing about this random screening, such approaches are very cumbersome. They are time consuming, they are expensive, they are inefficient to produce the fruitful results. Random screening we are discussing. And the drugs that are discovered through this random screening are morphine, atropine, digitalis, quinidine. These are the drugs which are discovered through the random screening. Penicillin was also discovered this way, which heralded the beginning of the antibiotic era or antibiotic therapy. Right? Cyclosporine, which is an immunosuppressant drug, which was discovered during the course of routine screening of the fungal products. So these are the drugs which are discovered through the random screening, right? Penicillin or cyclosporine or all these drugs. Now serendipity. Serendipity, or it is also called as discovery by chance. Sometimes new use of old drug is discovered. New use is discovered for an older drug, or its side effects find new therapeutic application. For example, lignocaine, which is a local anesthetic, phenytoin, which is an anti-epileptic drug. We can use them as an antiarrhythmic nowadays. Lignocaine, which is a local anesthetic, phenytoin, anti-epileptic drug, but we can use them as the antiarrhythmic drugs. Methotrexate is an anti-cancer drug, but it is also available for psoriatic treatment or for rheumatoid arthritis, right? Cyclophosphamide, azathioprine, these are the cytotoxic drug, but they are immunosuppressant and they are used for prevention of tissue rejection in the organ transplantation. So what we are discussing? We are discussing the serendipity, discovery by chance, right? Means new use is discovered for an older drug, or its side effects finds new therapeutic application that we have just discussed, like lignocaine, phenytoin. They were local anesthetics. Lignocaine is a local anesthetic and phenytoin is a anti-epileptic, but we can use them as a antiarrhythmic. So serendipity. Methotrexate, we know that it's an anti-cancer drug, but we can use methotrexate for the psoriasis or rheumatoid arthritis. Same thing with the cyclophosphamide, azathioprine. These are the cytotoxic drug, but because they are immunosuppressant, we can use them for the prevention of the tissue rejection in organ transplantation, right? Now, molecular modification of a known drug. Molecular modification of a known drug. In these procedures, what happen? Chemical analog of established drug are synthesized and tested for their biological activities to yield new compound. There is a molecular modification of a known drug, right? So which after some advantage in their pharmacokinetic and pharmacodynamic properties, or they have reduced toxicities. That means development of thiazide diuretics from much less used carbonic anhydrase inhibitor. So what we have done? We have done the molecular modification of a known drug. Many semi-synthetic penicillins and cephalosporin have been developed in this manner. We are only doing the molecular modification. Although it is a more logical approach, Yet, due to pressure from drug industry or their marketing promotion strategies, the molecular modification end up in a molecular manipulation. Means as a result, they so-called new drugs are virtually serving as a me too drugs. Means they have no added advantage over the already existing drug, and we call it as a drug explosion. Molecular modification of a known drug. So what we do in this procedure, chemical analog of established drug synthesized and tested for the biological activities to yield new compound. And this new compound is the molecular modification of a known drug, which offers some advantage over the pharmacokinetic and pharmacodynamic properties or reduced toxicities. Example is the thiazide diuretics from the carbonic anhydrase inhibitor, or many semisynthetic penicillins or cephalosporin have been developed through molecular modification. But this molecular modification of known drug is a logical approach. It is a logical approach because from pressure from the drug industry or their promotion marketing strategies, molecular modification mostly ended in a molecular manipulation, because they are so-called new drugs and virtually serving as a me too drugs, because they have means no added advantage over the already existing drug, and that may lead to the drug explosion. For example, we have omeprazole, pantoprazole, rabeprazole, lansoprazole, but they don't have any specific advantage. So all these drugs will consider as a molecular modification, or we can call them as a drug explosion or me too drugs. Now, rational drug designing. What is meant by rational drug designing? This procedure of drug discovery based on understanding of the biological activity and chemical structure. So, rational drug designing, development of AC inhibitors from the study of structure activity relationship of enzyme, active site inhibitors, availability of large number of H2 receptor blockers based on the H2 receptors, designing of a prodrug or an active metabolite. So basically what we are doing, rational drug designing. This procedure of drug discovery based on the biological activity and chemical structure. Means how will you develop AC inhibitors from the structure activity relationship of an enzyme. Means we know the enzyme, then we have discovered the AC inhibitors. So the drug discovery based on the chemical structure. Then availability of large number of H2 receptor blockers, ranitidine, cimetidine, famotidine, based on the characteristics of a H2 receptor. First H2 receptors are developed, then there is the development of H2 receptor blockers. So rational drug designing. So designing of a prodrug or an active metabolite. What is prodrug? Agent that is administered as a precursor of a drug, which is converted to its active metabolite or active therapeutic agent inside the body. So there is an unquestioned success of levodopa, which is a prodrug of neurotransmitter dopamine we use in the treatment of Parkinsonism. Besides prodrug, the active metabolites of certain drugs are also have therapeutic advantages over the parent compound. For example, paracetamol, an active metabolite of phenacetin, which is a safe analgesic. So designing of a prodrug or an active metabolite. What is prodrug? We know that. Agent that is administered as a precursor of a drug, which is converted to its active therapeutic agent inside the body. We know the unquestioned success of levodopa, where we use levodopa in the treatment of Parkinsonism, which is a prodrug of neurotransmitter dopamine. Besides prodrug, active metabolites of certain drugs are also have therapeutic advantage over the parent compound. Paracetamol, an active metabolite of phenacetin, which is a safe analgesic. Both are available, but paracetamol is most commonly used now. So these are the rational drug designing. Now, N-acetylprocainamide. N-acetylprocainamide, active metabolite of procainamide, which is local anesthetic. They act like an antiarrhythmic. N-acetylprocainamide, active metabolite of procainamide. This procainamide is developed from the procaine. So they act like a antiarrhythmic, no incidence of lupus-like syndrome with N-acetylprocainamide. After the synthesis or isolation of test drug, purity is ascertained by physicochemical and analytical studies. Promising lead compound is selected and they're subjected to the preclinical evaluation. Means after all these procedures, we select them or as a lead compound for the preclinical evaluation, right?